Assalamualaikum. Okay. So for today's lecture, okay, we are going to look at the next subtopic. So hopefully by the end of this lecture, we can cover all chapter 4 lah. Okay. All right. So for subtopic 4.5, which is cofactors, so we are going to look at some terms, okay, which is enzyme and holoenzyme. Okay, so basically some enzymes, okay, most enzyme, in order for the enzyme to work, okay, they have to have cofactors, okay, and these cofactors are located in the active site of the enzyme. All right, so the function of the cofactors is basically to assist in the enzyme reaction. Okay, so the first term that you have to know is enzyme. Okay, what is enzyme? Enzyme is the enzyme that is in the inactive state because it has no cofactors that can be found in the active site of the enzyme. Okay, so in order for the enzyme to be functional and activated, so cofactors has to bind to the active site of the enzyme and to activate the enzyme. Okay, so the complete and activated enzyme with its cofactor, we call it as holoenzyme. Okay, so enzyme without cofactors, so it is inactive. Holoenzyme is enzyme with cofactors and it is in the active state. Okay, so so this is a diagram to show you. Okay, so you have the enzyme, and then this is the allosteric site of enzyme. Okay, where inhibitor or activator will bind to the enzyme, and then here is the active site of enzyme where coenzyme is a type of cofactor. Okay, coenzyme is a type of cofactor, and then it will bind to the active site of the enzyme to activate the enzyme. Then only the substrate can enter and enzyme catalyze the reaction. So okay, so first you have to know what is cofactor. So cofactor is basically a non-protein molecule or ion needed for proper functioning of enzyme, and they are known as enzyme helper. So they will assist in the enzymatic reaction. Okay, so they are made up of either ions or some cofactors are made up of vitamin B. Okay, so those are cofactors that are found in the active site of enzyme. So many enzymes require cofactors. So the cofactor will help, okay, or assist in the enzymatic reaction. So by activating the enzyme, by altering the shape of the enzyme, or it can also participate in the chemical reactions. Okay. So cofactor may be bound tightly, okay, to the enzyme as permanent residents, or they may bind loosely and reversibly along with the substrate. Okay, so so if ions is the cofactor, they can bind to the active site of the enzyme tightly, okay, tightly to the active site of the enzyme, and they are permanently inside the enzyme. Okay. And as for coenzyme, they bind loosely and reversibly to the active site of the enzyme. Okay, so cofactor, you can divide cofactor into two types. Okay, which is organic or inorganic. Okay, so example of cofactor that is organic, which is coenzyme. Okay, coenzyme, and then the other one which is not included in the notes is you have prosthetic groups. Okay. So coenzyme involve in the transfer of chemical groups, atoms, or electrons. Okay, so example of organic cofactor, which is coenzyme, which is you have NAD plus, NADP plus, ATP coenzyme A. Okay, so basically these two molecules, okay, all these molecules you can find in the reactions, okay, that involve cellular respiration that we are going to look at in chapter 5 for NAD plus. for NADP plus is the cofactor that involve in one of the in several steps. Okay, in several steps of photosynthesis. Okay. So cellular respiration, photosynthesis involve many biochemical reactions to occur. It involves many enzymes, and this enzyme requires cofactor. And one of the cofactor is NAD plus and also NADP plus. Okay, so the cofactor NAD plus, okay, is a coenzyme for a number of dehydrogenase enzyme and act as hydrogen acceptor. So if you look at the name dehydrogenase enzyme, it means that the enzyme will remove, okay, remove hydrogen ion or atoms from the substrate molecule, okay, and then transfer to another molecule before it produces the product. So this is one of the reaction, okay, that involve NAD plus. Okay, so the enzyme here is malate dehydrogenase. Okay, remove hydrogen atom, okay, from the substrate. So the substrate here is the is malate. Okay, so malate. So this is the structure of malate. It is a 4-carbon compound. Okay, a 4-carbon compound. Basically, this is the reaction that that involve one of the step in cellular respiration. Okay, so malate is a 4-carbon compound. So what is the function of NAD plus? Okay, during the reaction, it is to remove 2 hydrogen atom from malate. Okay, so that's why the name of the enzyme is dehydrogenase. Okay, remove hydrogen atom. So who will remove the hydrogen atom? The cofactor, the coenzyme here. Okay, so it removes 2 hydrogen atom from the malate, from the substrate, and one of the hydrogen atom, the NAD plus will take up to form NADH. Okay, NADH, and another hydrogen atom or ions will be released to the surrounding solution. Okay, to the surrounding solution, and then the product will be oxaloacetate. Okay, so as you can see, there is no more hydrogen atom. So you have the formation of carbonyl group here. Okay, as the result of the reaction. So the product will be oxaloacetate, which is also a 4-carbon compound. All right. So the next type of cofactor is you have which is inorganic ions. Okay, so usually they are metal ions, such as, for example, you have calcium ion, zinc ion, magnesium, ferrum, chloride ion, and also cuprum ion. Okay, so metal ion, what is the function of this metal ion? It helps to draw electrons away from substrate molecules. So as you know, the function of electrons within an atom of a molecule is that it involves in the formation of covalent bond within the atom of that molecule. Okay, so when the ion pulls away the electron, it causes the bond within the molecule to be less stable and easier to break the bond. So that's why if you look at this statement, it also participates in the chemical reaction by drawing electrons away from the substrate, taking away hydrogen atom from the substrate. Okay, so that is how the cofactor participate in the chemical reaction. Okay. So example for calcium ion, where can you find calcium ion? It is the cofactor that is needed for blood clotting mechanism. Okay, so calcium ion are needed to activate thrombokinase, which converts prothrombin to thrombin in blood clotting. Okay. And then another ion, you have chloride ions. Okay, so this will increase the salivary amylase activity. Okay, so salivary amylase are the enzyme that are found in your saliva, which is secreted by the salivary gland to digest carbohydrates. Okay, the food that you eat. Okay, that contains carbohydrates. Okay, so that's for the cofactor. So now we are going to move on to the next subtopic, which is 4.6, which is enzyme inhibitions. Okay, so first you have to know what are inhibitors. So there are certain chemicals, chemical molecule that selectively inhibit the action of specific enzyme. Okay, it will inhibit the specific action of specific enzyme. Okay, only certain inhibitor will affect certain enzyme. It is like that. All right. So you can classify inhibitors into two types here, which is either reversible inhibitors or irreversible inhibitors. Okay, so for reversible inhibitors, they will form weak bonds such as hydrogen bonds with enzymes. Okay, so the interaction between the inhibitors and the enzyme is through the formation of weak bonds. Okay, so because the interaction is through the formation of weak bonds, this bond can easily be broken. Okay, the bond can easily be broken. So as long as the inhibitor are inside the enzyme, okay, are bound to the enzyme, so the enzyme cannot function or the rate of reaction becomes slower. But because the interaction is through weak bonds, this bond can easily be broken and the inhibitor can detached, okay, can be removed away from the enzyme. And so once the once the inhibitor is removed, the enzyme can function again to catalyze the reaction and the reaction rate will increase again, okay? So, so you can classify reversible inhibitors into two types, which is competitive and non-competitive inhibitors. Okay, so this is based on where they will bind to the enzyme. So for competitive inhibitors, they will bind to the active site of enzyme. Okay, active site. But as for the non-competitive inhibitors, they will bind to the allosteric site of enzyme. Okay, so remember that. And then, so for the next one is irreversible inhibitors. So they will bind tightly and permanently to the enzyme because the bond is tight. Okay, so this is due to the formation of covalent bonds. So as you know, covalent bonds is a strong bond, is a type of strong bonds. Okay, so when covalent bonds are formed between the inhibitors and the enzyme, so the interaction will be permanent. Okay, so it means that if irreversible inhibitors binds to the enzyme, it will permanently destroys the enzyme function. Okay, so maksudnya inhibitors ni bila dia bind je kepada enzyme tu, okay, so dia akan menyebabkan enzyme tu tak boleh berfungsi langsung. Tapi kalau reversible inhibitor boleh. Okay, sebab you can remove the inhibitor from the enzyme because the bonds because the interaction is through the formation of weak bonds. Okay, so reversible inhibitors, there are two types, okay, which is competitive and non-competitive inhibitors. So as we've mentioned, okay, so the interaction between the inhibitors and the enzyme is through the formation of weak bonds that can easily be broken. Okay, so examples, you have the formation of hydrogen bonds. Okay, and then the inhibition only takes place whilst the inhibitor is attached to the enzyme. So it means that the inhibitor can be removed. All right, so these weak bonds can easily be broken. And then you have here the diagram. So this is the normal reaction. Okay, so you have the enzyme and then there's no inhibitor present in the reaction, so the substrate can happily bind to the active site and reaction can occur as usual. But in the presence of competitive inhibitors, okay, which is competitive inhibition, so you have here the competitive inhibitors. Why do we call it as competitive inhibitors? Because the competitive inhibitors will compete, okay, with the substrate to bind to the active site. Okay, so once the competitive inhibitor binds to the active site, so substrate can no longer bind to the active site and the enzyme cannot catalyze the reaction. Okay, so that is the keyword, competitive inhibitors binds to the active site of the enzyme. As for the non-competitive inhibitors, it doesn't care about the active site. It will straight away binds to the allosteric site of the enzyme. But the effects is that once the non-competitive inhibitors bind to the allosteric site, it causes, as you can see from the diagram, the active site of the enzyme to change shape. Okay, the enzyme changes shape, and so therefore the active site also changes shape. So now the substrate cannot bind to the active site because the structure of the active site now is not complementary with the shape of the substrate. Okay, so that is non-competitive inhibitions. Okay, so the inhibitor binds to the allosteric site and causes the enzyme to change shape. All right. Okay, so so here are the detailed explanation for both inhibitors. So for competitive inhibitors, so they will have similar shape, okay, similar shape to the natural substrate. All right, close structural similarity. The shape of the inhibitors, they are not exactly the same, okay, because they are different molecule, inhibitor and also substrate. The keyword here is similar, lebih kurang sama. Okay, lebih kurang sama. So that's why inhibitors can bind to the active site. So do not use substrate and inhibitor have the same shape. So that is not correct. Okay, they have similar shape or they mimic each other. Okay, the inhibitor mimics the substrate. Pun boleh juga. Alright. So because the shape of the inhibitors is similar to the substrate, so the inhibitor will compete with the substrate to bind to the same active site, and so this the effects is that the rate of reaction will slow down, and then the product produced will be lesser. And then, for competitive inhibitors, you can overcome, okay, the effects of competitive inhibition by increasing the substrate concentration. What is the advantage of increasing the substrate concentration? Because you will have more substrate in the reaction. When you have more substrate in the reaction, there will be higher tendency for the substrate to bind to the active site instead of the inhibitor. Okay, so more substrate molecule you have to add in order to increase or to overcome the effects of competitive inhibition. So here you have a diagram here, okay, to show an example of competitive inhibition. So the enzyme is succinate dehydrogenase. Okay, so here is succinate dehydrogenase, and the substrate here is the succinate. Okay, so and the product formed, okay, the succinate binds to the active site, reaction occurs. You have the formation of succinate enzyme complex, which is enzyme substrate complex here. Okay, and then the product will be formed, which is fumarate and also the enzyme. But in the presence of competitive inhibitors, okay, which is malonate, as you can see, the structure of malonate is similar to the succinate. Okay, so because the structure is similar, so malonate will compete to bind to the active site. Once the malonate binds to the active site, succinate cannot bind and reaction cannot occur, or reaction rate reduce, okay, and no product form or less product will form. Okay, so the next one is for non-competitive inhibitors. Okay, so for this one, the shape of the inhibitors is totally different from the substrate. Okay, it's totally different. So the inhibitor will not compete with the substrate. It does not compete with the substrate to bind to the active site because it binds straight away to the allosteric site of the enzyme. Okay, so the effects of the binding to the allosteric site is that this will cause the enzyme to change its shape or conformation, and causes the active site to also change shape. Okay, so once the active site changes shape, the substrate cannot fit into the active site, and so therefore reaction cannot occur. Okay, so prevent substrate to bind to the active site. So enzyme will not function as long as you have the inhibitor binds to the allosteric site. Okay, but this type of inhibitor, the inhibitor can be removed. Okay, as long as the inhibitor is inside the allosteric site, reaction cannot occur. But once the inhibitor is removed, then the enzyme can function again. Okay. So that is for reversible inhibitor that of non-competitive inhibition type or inhibitors type. All right. Okay, so this graph, it shows you the rate of reaction for three different types of reaction. Okay, so the y-axis is for rate of reaction or velocity, okay, of the reaction, and the x-axis is substrate concentration. So you have three graphs: the green graph, the blue graph, and also the red graph here. Okay, so for the green graph here, as you can see, the rate of reaction occurs really fast, okay, and reaches maximum velocity really quickly. Okay, so this is for the normal rate of reaction without the presence of any inhibitors. Okay, normal reaction without any inhibitors. Rate of reaction occurs really fast and reaches maximum velocity really quickly. But for the blue graph here, okay, in the reaction, you added the competitive inhibitors, okay? So what is the effects of adding competitive inhibitors to the reaction? The rate of reaction becomes slower and less product will be produced, less product, right? But as you add more substrate concentration to the reaction, okay, so somehow the rate of reaction will also increase, but at a slower rate. But it will reach the maximum velocity as the natural reaction, okay, without the inhibitors. So this is how you overcome competitive inhibitors, by increasing the substrate concentration. But for the red graph here, okay, it shows you the rate of reaction in the presence of non-competitive inhibitors. Okay, non-competitive inhibitors. So the rate of reaction slows down and stops. Okay, so there's no effects of you adding more substrate. Okay, so because the inhibitor will just bind to the allosteric site, and no substrate can bind to the active site. Okay, so there is no point of you adding substrate concentration. So that is for the red graph here, non-competitive inhibitors. So you have to know the difference, and you have to know how to explain this graph. Okay, pernahlah keluar dalam exam. Kalau dalam awak tengok dalam buku latihan awak pun disuruh lukis graf kan, lukis graf untuk rate of reaction, okay, in the presence of inhibitors. So you have to know how to do the graph, and also you have to know how to explain this graph. Okay. So the next one is irreversible inhibitors. So irreversible inhibitors or non-reversible inhibitors, they will bind tightly and permanently to the enzyme with strong covalent bonds, okay, tightly through the formation of covalent bonds between the enzyme and also the inhibitors, and this will destroy the enzyme function. So this can occur at low inhibitors concentration. So if you add, as soon as you add the inhibitors to the reaction, the rate of reaction will straight away slow down, okay, or even stops. So here, saya nak kena padam sikit ni kot. Tak boleh padam. Sekejap. Right. Okay, so example of irreversible inhibitors, you have toxin and also poisons. Okay, toxin and poisons. So example of toxin here, which is sarin. Okay, so sarin is a nerve gas. All right, so it has no odor, no taste. It will target the nervous system. So it says here, the serine molecule will bind covalently to the R group on the amino acid serine, which is located in the active site of this enzyme, acetylcholinesterase, okay, which is a nervous system enzyme. So once the serine molecule, okay, binds to the active site of this enzyme, it causes the enzyme to change its shape, okay. So now the enzyme cannot bind and break down its substrate, which is acetylcholine. So acetylcholine is the substrate for the enzyme acetylcholinesterase. What is acetylcholine? It is a type of neurotransmitter. Okay, so you have to know first what is the function of this acetylcholine. The function of acetylcholine is to allow impulse, okay, to be transmitted from one neuron to another neuron to another neuron until the impulse reaches muscle cell and causes the muscle cell to contract. Okay, so that is the function of acetylcholine. But you do not want continuous transmission of impulse to your muscle and causes your muscle to always contract and can lead to spasm, muscle spasm. Okay. So, so this acetylcholine has to, in order to stop transmission of impulse, this acetylcholine has to bind to this enzyme, okay, to break down this neurotransmitter into smaller molecule and causes and there will be no neurotransmitter in the synaptic cleft to transmit the impulse. Okay. But when acetylcholine cannot bind to the enzyme, here acetylcholine cannot be broken down and it will cause the acetylcholine to accumulate in the synaptic cleft. Okay, synaptic cleft is the region or the space between two neurons, okay, two neurons that allows the impulse to be transmitted. Okay. So the nerve impulse cannot be stopped, causing continuous contraction of the muscle. So this will lead to convulsion, paralysis, and also eventually death. Alright. So dia kalau tengok kat sini, okay. So for example, we have the first neuron. Okay, here you have the first neuron. Okay, so this is the cell body. This is the dendrite. Okay, tak berapa nak cantik saya lukis. Alright. So this is the axon. So this is the synaptic terminal. Alright. So this is the first neuron. Second neuron. Okay, the same thing. The cell body. Tak apa nak cantik, sorry. Okay, and then axon. And then axon terminal. Alright. And then so the target cell here is the muscle cell. Muscle cell. Okay. So impulse. In order to transmit impulse from neuron 1, okay, to neuron 2, the first neuron has to release neurotransmitter acetylcholine in the synaptic cleft. Okay, so you have acetylcholine in the synaptic cleft. The function of this acetylcholine is that it will bind to the receptor on the dendrite of the second neuron. Okay. So once this acetylcholine binds to the receptor on the dendrite of the second neuron, then only impulse can be transmitted. Okay, impulse can be transmitted and then received by the muscle cell and causes the muscle cell to contract. But you do not want continuous transmission of impulse and causes continuous contraction of the muscle cell. So this acetylcholine has to be removed. In order to remove this acetylcholine, you will need the enzyme acetylcholinesterase to break down the neurotransmitter. Okay. So, but in the presence of serine, okay, so serine will bind to the active site of acetylcholinesterase permanently, okay, and destroys the function of acetylcholinesterase, and so therefore acetylcholine will accumulate in the synaptic cleft and causes continuous contraction of the muscle. Okay, so that is the effects of serine. Okay, it causes acetylcholinesterase cannot break down the acetylcholine because serine binds to the active site of the enzyme. So that is the effects of serine. Kita panggil dia serine sebab dia binds to the amino acid serine, which is located on the active site of acetylcholinesterase. Something lagi ada kat sini. Okay, so far do you have any question on this part?
Ya.
Yeah. Alright.
Tak ada, madam.
Faham. So saya boleh proceed pada the next subtopic, which is 4.7, which is regulation of enzyme activity. So when reaction occurs and at what rate. So basically for this slide, it gives you several examples, okay, of how enzyme activity is regulated. Okay, several example, several situation of how enzyme activity will be regulated. Okay, so where do you find enzymes? Enzymes are found in living cells. And what is the function of enzyme? It is to catalyze metabolic pathway within the cell. What is metabolic pathway? Biochemical reactions that occurs inside cells. Okay, biochemical reaction that occurs inside living cells, and it requires several enzymes in the reaction to produce the products. Okay, several enzymes, not only one, but several enzymes in the reactions. So how to regulate the enzyme activity? So these are several examples, okay, how enzyme activity are regulated. The first one, genes that codes for enzyme may be turned on or off. Example, gene that codes for beta-galactosidase is activated when lactose is present. For this example, okay, it means that, okay, so in order to synthesize the enzyme beta-galactosidase, okay, as you know, enzymes are protein. So how does protein is synthesized? It is synthesized based on the information coded in the genes. Okay, the protein, the enzyme here is synthesized based on the gene that is coded. Sorry, based on the information that is coded in the genes. Okay, so in order for the gene to synthesize the enzyme, the gene has to be turned on or off. Okay, so turn on or off means that the gene at some situation it is turned on, at some situation it is turned off. So turn on, it means that the gene can be used to synthesize the enzyme. If it's turned off, it means that the gene cannot synthesize the enzyme. So how to turn on in this instance, for this example, how to turn on the gene? So in the presence of lactose, okay? So when lactose is present inside cell, then the gene is turned on to synthesize the beta-galactosidase. Basically, for this enzyme, the function of beta-galactosidase is to break down lactose into glucose and also galactose. Okay, the monomers for lactose is glucose and galactose. Okay, so that is how you turn on genes to synthesize enzyme. Okay, you have certain situation that will turn on the gene, or certain situation that will turn off the gene. In this case, the presence of lactose will turn on the gene to synthesize the enzyme beta-galactosidase to break down the lactose. Okay. So yang ini sebenarnya salah satu contoh yang kita akan belajar dengan lagi detail semester 2 nanti dalam chapter genetik. Ini just a statement. Just a statement yang kita akan belajar dengan lebih detail proses dia masa chapter semester 2. Okay, chapter genetics. Okay, for second example, inactive enzymes such as pepsinogen. Okay, so some enzyme, okay, so enzyme when it is synthesized, it is in the inactive form. Okay, so example you have here is pepsinogen. Pepsinogen is enzyme that is in the inactive form. It cannot function. So how to activate this enzyme? Okay, so this enzyme basically are produced, okay, by cells in the stomach. Okay, your stomach will produce pepsinogen. So your stomach also will produce hydrochloric acid. Okay, so in order to activate the pepsinogen, the hydrochloric acid will activate the pepsinogen to become pepsin. Okay, so pepsin. So pepsin asalnya adalah pepsinogen. So sebab tu awak punya stomach kena hasilkan hydrochloric acid to activate pepsinogen. So hydrochloric acid has other function actually, okay, which is to destroy bacteria, okay, that enters into your body, into your stomach from the food that you eat. Okay, so that is one other function of hydrochloric acids, to kill bacteria. And then okay, so that is one example. Okay, so the enzyme is activated in the presence of other molecule. In this case is hydrochloric acids. Okay, And then the next one is the presence of inhibitors such as reversible inhibitors or reversible inhibitions. Okay. So so for normal reaction occurs and the rate of reaction increases. But as soon as you add inhibitors to the reaction, the rate of reaction will slow down. Okay, and then the next one is a regulator molecule. Okay, such as activator or inhibitors. Okay, so if activator binds to the enzyme, it will activate the enzyme. If inhibitors bind to the enzyme, it will inhibit the enzyme. So this one we call it as allosteric regulators. You have two types, activator and also inhibitors. Okay. So because the name is allosteric regulators, so they only bind to the allosteric site of the enzyme and causes the enzyme to change shape. Okay. So either in the active state or in the inactive state. Okay. So for the last example here, the end product may inhibit enzyme, which is feedback inhibition. So feedback inhibition is a type of mechanism or process that will regulate enzyme activity. So in this reaction, in the feedback inhibition reaction process, okay, so the metabolic pathway, you have the initial substrate, okay, and then you have the first enzyme, and then catalyze the reaction to produce intermediate product, and then you have the second enzyme produces intermediate product, the third enzyme produces intermediate product, and then the last enzyme produce the end product. Okay, end product. Okay, so this if the reaction continually occurs, okay, so the concentration of end product will increase. Okay, so you have an accumulation of the end product. So you want to stop the reaction for a while. So therefore, the end product will become in the inhibitors. Okay, the end product will become the inhibitors. Saya short form je lah. Okay, inhibitors for the first enzyme of the reaction. Okay, so this end product will become inhibitors for the enzyme, for the first enzyme of the reaction, and this end product will bind to to only the allosteric site of the first enzyme. Okay, usually the first enzyme. So that is the explanation for example number 5. Okay, end product will become inhibitors for usually for the first enzyme of the reaction. Okay. Padam eh. Padam dulu sekejap, sorry. Okay, so allosteric regulations. Okay, so allosteric regulation. So what? So for this allosteric regulation, it involves regulatory molecules. Okay, so you have two types of regulatory molecule, which is activator or inhibitors. Okay, so regulatory molecules will bind briefly and reversibly to another site. That's why we call it as allosteric regulation, to another site, which is the allosteric site of the enzyme, and causes the enzyme to change shape and function. Okay, so function either in the active state or in the inactive state. Okay. So you have two types of allosteric regulation, which is the first one, which is allosteric activation. The second one is allosteric inhibition. So for allosteric activation, allosteric regulators, which is the activator, will bind to the enzyme and causes the enzyme to be activated. Okay, activating the enzyme. But for the allosteric inhibition, so it is a form of non-competitive inhibitions, okay, because the inhibitor will bind to the allosteric site of the enzyme and causes the enzyme to change shape into the inactive form, okay, into the inactive form. Okay, so this slide I've added to explain to you for this type of allosteric regulation, it will involve allosteric enzyme. Okay, so what is allosteric enzyme? Kalau tengok nanti kan, kalau dekat slide tahu ada enzyme, alright, enzyme that has 2 or more subunits. Okay, 2 or more subunits. So this is an example. Okay, enzyme with 2 or more subunits. So one subunit with its active site, the second subunit with its active site, second, third, and the fourth. Okay, this one, it has 4 subunits. Okay, for instance. So each of these subunit, they are the same enzyme. It means that they will bind to the same substrate. Okay, they have the same active site, binds to the same substrate. So each enzyme oscillates between active and inactive form. So the characteristics of this allosteric enzyme is that it is not stable. Okay, it is not stable. It will keep on changing from the active form to the inactive form, active form, inactive form. So how to maximize the enzyme activity rate or reaction is that you have to stabilize the enzyme first. Okay, you have to stabilize the enzyme first, either in the active form or in the inactive form. Okay, so it says here each subunit has its own active site and allosteric site. So this is the active site. Okay, this is the active site where the substrate will bind. But where the subunit joins, okay, where the subunit joins here, so this usually will become the allosteric site. Okay, where the subunit joins, usually it becomes the allosteric site where the regulatory molecule will bind to. So regulatory molecules, there are 2 types, activator and inhibitors that binds to the allosteric sites. Okay, often located where subunit joins. So binding by regulatory molecule will cause the enzyme to change shape. Okay, either in the active form or in the inactive form. Okay, active form activate the enzyme, inactive form will inhibit the enzyme activity. Okay, so this is the enzyme. Okay, so it has, this is the allosteric enzyme. Okay, allosteric enzyme. So as you see, the allosteric enzyme, the molecule is not stable. Okay, it will keep on changing from the active form to the inactive form, active form, inactive form, continuously. But you have to stabilize the enzyme. Okay, so if you add activator to the reaction, the enzyme will be stabilized in the active form. But if you add inhibitor to the reaction, it will stabilize the inactive form of the reaction. And in order to stabilize the active form of the enzyme or the inactive form of the enzyme, the enzyme only requires 1 molecule of activator or 1 molecule of inhibitor. So when 1 molecule of activator binds to one of the active site, it causes all of the subunit to be in the active form. Okay, all. Satu je diperlukan. Satu molekul activator je diperlukan untuk activatekan semua subunit. Ataupun satu je inhibitor diperlukan untuk bind kepada allosteric site dan menyebabkan all subunit of the enzyme to be in the inactive form. Okay, so that's how regulatory molecule works. Okay, so the next tadi allosteric regulation. Okay, allosteric regulation yang melibatkan allosteric regulators. Okay, the other mechanism is cooperativity. Okay, so for cooperativity, it also involves allosteric enzyme. Okay, so as you know, allosteric enzyme, they are not stable. So in order to stabilize the enzyme in the active form, okay, so for cooperativity, this substrate will become the activator, okay, that activates all the subunit. So for cooperativity, you only need 1 substrate, okay, 1 substrate here to bind to one of the active site and causes all the subunit to be locked in the active form, okay, to be stabilized, all active site to be stabilized in the active form. So that is cooperativity. Kalau regulatory allosteric regulation, dia melibatkan allosteric regulator. Tapi kalau cooperativity, dia melibatkan substrate that will become the activator that binds to the active site and causes all subunit to be activated. Okay, so that is cooperativity, a type of allosteric activation. So this mechanism amplifies the response of enzyme to substrate. Okay. So binding of a substrate molecule to one active site causes the second and the subsequent substrate to be to bind more readily. To bind more readily means that it is in the active form. Okay, so locks one substrate locks all subunit in the active form. Okay. Okay, so the third regulatory mechanism of enzyme action is feedback inhibition. Okay, feedback inhibition. So ini yang ketiga lah. Yang kedua cooperativity. Yang pertama adalah allosteric regulation. So 1, 2, okay, and 3. The third one. Okay. So feedback inhibition, activity of an enzyme near the beginning of a metabolic pathway is inhibited by the end products. Okay, near the beginning of a metabolic pathway, it means that it usually involves the first enzyme. Okay, usually it involves the first enzyme. So it is a type of allosteric inhibition because the end products will become the inhibitor that binds to the allosteric site of the first enzyme. So example, accumulation of isoleucine allosterically inhibit the enzyme in the first step of the pathway. First step of the pathway, it involves the first enzyme. Okay, metabolic pathway is a reaction that occurs inside cells that involve many enzymes. Okay, several enzymes in the reaction. So what is isoleucine? Isoleucine is the end products. Okay, isoleucine is the end product that will become the inhibitor for the first enzyme. So what is the significance of feedback inhibition? It is to prevent, prevents the cell from wasting chemical resources to synthesize more isoleucine than is necessary. So maksudnya apa kepentingan ataupun kebaikan feedback inhibition ni supaya nak memastikan sel ni tak waste chemical resources. Sel tu hanya akan menghasilkan end product pada kadar yang sel perlukan. Okay, so bila dia dah hasilkan enough end product, so the production of the end product akan stop sekejap. Okay, biarkan sel tu guna end product tu dulu. So bila end product concentration reduced, then only the end product akan keluar daripada allosteric site of the first enzyme, and then the first enzyme can function again to catalyze the reaction again to produce the end product again. Okay, so this is a diagram. Okay, so you have here the first enzyme. Okay, threonine deaminase. Okay, threonine deaminase is the first enzyme. So this the substrate for threonine deaminase is threonine. Threonine and isoleucine is a type of amino acid. Okay, so this chemical reaction or metabolic pathway is an example of metabolic pathway that occurs inside E. coli. Okay, so E. coli will synthesize isoleucine from threonine. Okay, for cell to use, for the E. coli to use. Okay, so the first enzyme binds to the threonine and causes a series of reactions to occur that involve several enzymes. Okay, before the end product is produced, which is the isoleucine. So the end product, when the concentration is high enough, okay, it will cause the isoleucine to bind to the allosteric site of the first enzyme here, okay, threonine deaminase. But it will bind to the allosteric site and causes the enzyme to change shape, and threonine cannot enter the active site and the reaction is stopped. Okay. So this is to allow the isoleucine to be used by the cell first. Okay, so once the concentration of isoleucine has reduced, okay, so then isoleucine will be removed from the enzyme, and the enzyme can catalyze the reaction again, okay, to produce more isoleucine for the cell. Okay, so so far ada soalan tak untuk so far yang kita tengok ni?
Tak ada, madam. Okay.
Okay, faham So the next, kita move on to the next subtopic, which is 4.8, okay, classification of enzymes. Okay, so as you know, most enzyme, you can identify most enzyme by the name of the enzyme. Okay, usually they will end with the with ACE. Okay, usually they will end with ACE. So ada je jumpa ACE, maksudnya itu adalah enzyme. Okay. So the enzymes are classified into 6 classes, okay, based on the type of reaction the enzyme catalyzes, okay. So 6 classes. So you can classify the enzyme based on how the enzyme involved in the reaction. So you can have oxidoreductase, for example. So this type of enzyme involved in the oxidation reduction reaction. Okay. So daripada nama enzyme kelas tu, awak boleh tahu apa peranan ataupun macam mana tindak balas kimia tu berlaku. Okay. So this classification is based on recommendations of nomenclature Committee of the International Union of Biochemistry and Molecular Biology in 1964. Okay. Ini nama ni tak pernah tanya lah, awak tak payah hafal. Kita tak pernah tanya pun dalam exam. Okay, so just you have to know the 6 classes and how they involve in how the enzyme involved in the reaction. Okay, so for the first class you have oxidoreductase, the first class, and then you have transferase, and then you have hydrolase, lyase, isomerase, and ligase. Okay, so those are the 6 classes of enzyme. So for my slides, okay, I've added, okay, a few reactions to show you how the enzyme involved in the reaction. So for the oxidoreductase, it involved in oxidation reduction reaction or redox reaction. Okay, so as you know, the concept of redox reaction, it involves the transfer of electrons, the transfer of hydrogen atoms, because in the hydrogen atoms you have electrons. Okay, hydrogen ion, kan hydrogen ion. Kenapa dia jadi ion? Sebab apa? Dia akan riliskan electrons. Okay. So and also oxygen atom. So when it involves the removal or gain of these items, okay, oxygen, hydrogen, electrons. So it involved in the oxidation reduction reaction. So they are oxidoreductase. So example macam tadi lah, you have the enzyme malate dehydrogenase. D remove hydrogen atoms, okay, from the substrate. Here is the malate, and then here is the cofactor, okay, which is the coenzyme NAD+. So the NAD+ will remove 2 hydrogen ions, okay, or atoms from the malate, and malate will convert into oxaloacetate. Okay, so NAD+ become NADH, and hydrogen ion is released into the surrounding. So example, you have NADH dehydrogenase. And then for the second class, you have transferase. Nama dia nampak? Transfer. Transfer apa? Transfer functional groups. So functional groups kita dah tengok kan masa first chapter. Okay, so you have 7 functional groups. Okay, so example, you can have here hydroxy group or amino group. In this case is phosphate. In this case of example of this reaction is it involves phosphate group. Okay, so the enzyme involved here is hexokinase. Okay, so hexokinase is the enzyme involved in this reaction. So this is a reaction that you will see in chapter 5 many times. Okay, so this also a reaction that involved in cellular respiration. Dua-dua reaction ni adalah reaction yang awak akan familiar bila kita tengok chapter 5 nanti. Okay, so hexokinase, what is the function of hexokinase? It is to remove one phosphate group, okay, from ATP, and the phosphate will be given to glucose. So glucose will become glucose 6-phosphate because the phosphate is attached to carbon number 6 of the glucose molecule. Okay. So because one phosphate is removed from ATP, so ATP will become ADP. Adenosine triphosphate becomes adenosine diphosphate sebab satu dah bagi dekat glucose. Okay, so that is transferase involved in the transfer of functional group. Okay, so the next class is hydrolase. Okay, hydro apa maksud dia? Air, lyse mean pecahkan ikatan. Okay, so for this hydrolase, it involved in the reaction that breaks bond with the addition of water molecule. Okay, break bonds with the addition of water molecule. Hydrolase. Hydro air, lase, lysis, pecah. Okay. So example, you have dipeptidase, lipase, amylase, sucrase and peptidase. So this is a normal reaction. Yang ini kita tengok masa chapter 1 hari ni kan, tindak balas ni. Sucrose, water, and then the enzyme is sucrase to produce glucose and fructose. Glucose and fructose is the monomer for sucrose. So in order to break sucrose into monomer, you have to add water, okay, to the reaction to produce glucose and fructose. Okay, so the next one is lyse. Okay, so dia pendek daripada nama hydrolase ni. Ni lyse. Okay, so this enzyme will involve in breaking of chemical bonds without the addition of water. Tak ada hydro kat depan ni kan. So dia lyse je. To break chemical bonds without addition of water, ataupun it involve in the formation or removal of double bonds with group transfer. So example, you have fumarase, peptidase, PEP carboxylase. So example is pyruvate decarboxylase. Nampak? Dia terlibat dalam group transfer kan? So decarboxylase remove carbon, carbonyl group. Okay, carbon from the reaction. Okay, decarboxylase. So pyruvate is the substrate. Okay, so the function of pyruvate decarboxylase ni is to break pyruvate into two molecules, which is ethanol and carbon dioxide. Okay, so pyruvate is a 3-carbon compound. They break into 2 carbon and 1 carbon compound. Okay, so break bonds without the addition of water ataupun melibatkan group transfer. Okay, decarboxylase, remove carbon from the molecule. Okay, so the next class is isomerase. So nama pun boleh tahu isomerase. To catalyze the conversion of one isomer to another isomer, or catalyzes the rearrangement of atoms within a molecule and convert one isomer to another. So example here is triose phosphate isomerase. So this is the sambungan daripada tindak balas ni. Okay, glucose 6-phosphate. So daripada glucose 6-phosphate dia akan ditukarkan jadi fructose 6-phosphate. As you know, glucose and fructose are isomer of each other. So the function of phosphoglucoisomerase is to convert glucose to fructose. Okay, so glucose 6-phosphate becomes fructose 6-phosphate. And then the last one is ligase. Ligase daripada perkataan ligate. Okay, what is ligate? Ligate is to join two molecules together and then the process involve formation of bonds. Okay, so joining of two molecules using energy derived from the breakdown of ATP. Example is DNA ligase. So example, one of the step in protein synthesis. Ini step ni kita akan tengok masa chapter semester depan lah, semester 2. So this is an example. So you have the enzyme amino acid tRNA synthetase. Okay, so it means that this enzyme, okay, will catalyze or will join together tRNA dengan amino acid untuk jadi amino acid tRNA complex. Okay, so the reaction involve ATP and then ATP will release 2 of its phosphate. Okay, jadilah AMP, adenosine monophosphate, sebab dia dah release 2 phosphate. PI ni maksudnya inorganic phosphate. Okay, so you have so the reaction will ligate these two molecules together. Okay, will join these two molecules together. Okay, so the last subtopic is the application part, okay, which is the use of enzyme in the industry, okay, and also medical use. So you have this type of enzyme, you have protease, ligninase, glucose isomerase, lactase, and also trypsin. So for protease, they are used in the baking industry to break down proteins in flour for biscuit production, okay? And then ligninase in the paper industry to remove lignin from wood. So wood, by removing the lignin, you can produce paper. Okay, and then glucose isomerase, glucose isomerase. So involved in the food process industry, okay, processed food. And then for this one, glucose is to manufacture fructose syrup from glucose, okay. Kalau tengok air tin yang awak minum tu kan, air tin tu semua, dia dimaniskan oleh fructose syrup, okay. And then the next one is lactase, okay, in the milk production. So production of lactose-free milk, okay. So there are people that are lactose intolerant. Maksudnya bila dia makan, dia akan produce reaction, okay, allergic reaction dekat situ. So so for these people, in order for these people to take milk, so they will take lactose-free milk. So the function of this lactase is to break lactose into its monomer, which is glucose and galactose. So maksudnya susu ni hanya akan ada glucose and galactose je, dia tak ada lactose. Okay, so maksudnya susu ni akan jadi lebih manis lah. And then the next one, the last one is trypsin for medical use. Okay, so it involves in removal of blood clots and in wound cleaning. So for point 9, enzyme technology, the last subtopic. Okay, so enzyme, for enzyme technology, so enzyme can be extracted from cells, okay, used to catalyze wide range of commercially important processes. So some new processes developed to manufacture bulk and high added value products. So in the industry, you want to produce the products in large quantities that are more valuable, okay, that has added value. Macam tadilah nak hasilkan apa, susu yang lactose free kan, untuk hasilkan apa, roti yang lebih fluffy contohnya kan, untuk hasilkan kertas kan. So untuk hasilkan dalam industri, untuk hasilkan dalam kuantiti yang banyak dan yang lebih berkualiti lah. Okay, so so for this purpose we will use, okay, immobilized enzyme. Okay, so what is immobilized enzyme? Daripada nama tu boleh tahu apa keadaan dia. Immobilized, cannot move. The enzyme cannot move. So enzyme is attached or retained within insoluble or inert support. Okay, so maksudnya untuk keadaan, untuk immobilized enzyme ni, you have the inert support. Okay, a material or a support that you will attach the enzyme to. Okay, so you attach the enzyme to the support and now the enzyme cannot move. Okay, so the only thing that will move is the substrate. Okay, the substrate will move and find the enzyme and produces the product. Okay, so the product you can easily isolate from the reaction. So the product is not mixed with the enzyme and you can easily isolate the product from the reaction. So you can extract quickly, okay, the product from the reaction. So what is the advantage of immobilized enzyme? So the enzyme can be reused and continuous production. Okay, so you add substrate, product produced, product is isolated. Add more substrate, product produced, and the product is isolated. So you can easily isolate the product from the enzyme, and the enzyme can be reused over and over again to produce more products. Okay, so the enzyme does not contaminate the products. Okay, so maksudnya bila dalam reaction tu awak dah siap-siap ikat enzyme, so maksudnya tak adalah, senanglah awak nak asingkan produk daripada enzim. Tak adalah dia bercampur kan. So bila dia tak bercampur, awak senanglah nak asingkan produk daripada enzim. And then because the enzyme are tied to the support, so the enzyme are generally more stable. Okay, but the disadvantage of immobilized enzyme is that it can cause possible or partial loss of activity. Okay, because the reaction only relies on the movement of the substrate and not enzyme. Okay, so the activity will the reaction you will some of the reaction activity will be lost. Okay, so maksudnya kena apply more heat to the reaction. Okay, more heat to the reaction so that the substrate will move quickly to the enzyme. So this will change the kinetic of the enzyme. Okay, this will change the kinetics of the enzyme. Okay, so for this reaction you may apply thermal heat, and then this thermal heat can cause the enzyme to denature. Okay, or change the kinetics of the enzyme. Okay, so methods of immobilized enzyme. You have several methods. The first one is adsorption, and then you have entrapment, and then you have binding. So binding, you can have covalent bonding, cross-linking, and then the last one you have encapsulation. Okay, so this is how the enzyme bind to the support. Okay, so for entrapment, for adsorption, so enzyme is attached to the outside of an inert material. So this is the inert material, the support. Okay, the inert material that you attach the enzyme to. Okay, so the attachment to the support is through the formation of weak bonds, such as hydrogen bonds, van der Waals forces, hydrophobic interaction, and also ionic interactions. Okay. So formed between enzyme and insoluble or inert support. So the material for the support can be made up of ion exchange resin or hydrophobic resin. Okay. So for the next method is entrapment. Okay. So you trap enzyme in a 3D lattice. Yang ni. So these are the support. Dia macam satu bekas lah kan. Okay, the support. So where it has many compartments. Okay, many compartments that you place the enzyme into. Okay, and then you add the substrate into the compartment. Okay, and then reaction occurs, and this support only allows substrate and products to pass through, but not enzyme. Okay, so when reaction occurs, product can produce, so substrate can pass through. Product can pass through, and you can isolate the substrate from the enzyme. Okay, so enzyme are trapped within a confined space or network. So for this entrapment, enzyme are not chemically bound to the support. Kalau yang ni ada kan, ada formation of bonds, but this one there's no formation of chemical bond at all. So you just place the enzyme inside the support. So the support is made up of polymers such as alginate and also silica gel. So yang ini sekejap lagi saya explain, but this one first. Okay, binding. So the binding, you have two types, okay, which is through the formation of covalent bonds or cross-linking. So covalent bonding between the enzyme and the support material. So the support material is cellulose and collagen. Sama macam tadilah, lebih kurang kan. Dengan yang ni, dengan adsorption ni. Adsorption, weak bonds. Tapi kalau binding through the formation of covalent bonds. So you have the enzyme, you have formation of covalent bonds between the enzyme and the support. Okay. So support is made up of cellulose or collagen. Okay. So the next one is cross-linking. Enzymes are covalently bonded to each other. The support is made up of glutaraldehyde that acts as a protein cross-linking agent. So you have enzyme 1, enzyme 2, enzyme 3, enzyme 4, 5, and so on. Kan? So these enzymes are linked together. So that's why we call it as cross-linking. Okay, they are linked together, and the substance that links the enzyme together are made up of this material, glutaraldehydes. Okay, which is the protein cross-linking reagent. Okay, so for this type of method, okay, enzyme can be denatured due to bonding. Sebab dia melibatkan ikatan kan. So this type of method can denature the enzyme. And then the last one is encapsulation. So enzyme wrapped in semi-permeable membrane. So the membrane are made up of nylon or cellulose nitrate. So you have the support material that acts as macam capsule. Okay. and you place many enzymes inside the capsule, okay, for the reaction to occur. Okay, so these are examples of immobilized enzyme. Cross-linking, can you cross-link the enzyme together? This can cause the enzyme to denature, so that is the disadvantage. And then covalent bonding, okay, between the enzyme and the support, entrapment, encapsulation, and also the last one, absorption. Okay, end of chapter 4.